Hello and welcome to this lecture about bird populations as part of the basic course in ornithology. We'll begin this lecture with defining population. So what is a population? A population is essentially all the individuals belonging to a certain species that you're interested in that occur in the same place at the same time. So as an example, here we see part of the population of bar-headed geese that winters every year in Pongdam region. So when we are talking about this definition, there are three key elements that we should keep in mind. One is that it refers to all the individuals that belong to the species that you're interested in. So everything is taken into account. Second part it refers to is a certain area of interest. So you have to define that space within which you are talking about the population. So this area, if you are referring to the breeding season for a bird, it means that individuals of the species can breed during that time and through the process are able to contribute to increasing the size of the population. So this concept of containing the definition of or population to a certain area, we refer to it as geographic closure. Similarly, we also make a reference to temporal closure when we are defining a population. So it means that we are talking about these individuals which are occurring in that area during a certain time period, which we also define when we are talking about a certain population. So you must keep these three things in mind when you are defining a population in any ecological study. An important property of population is that it changes with change in time, and these changes in population are result of four basic processes, and these processes are births, which add to the population. Then there are immigration, which are the movement of individuals from outside this area that we are talking about when we are referring to our population of interest. Then there are deaths, which remove from the population, as well as emigration, which also remove from the population, because immigration is the process where individuals of the population that we are studying, they are moving outside this area that we are making a reference to. Births and immigrations obviously add to the population size, whereas deaths and immigration cause a decline in the population size. We also should remember that births and deaths are internal changes which are caused by processes that are happening with the initial population that we start with, and immigration and emigration are external changes or represent migration, which are caused only when the population is geographically open and allows movement of individuals from both within and outside this population that we are making a reference to. It is very easy to remember these four processes which cause changes in the population using this simple acronym BIDE, which refers to births, immigration, deaths, and emigration. And we can mathematically represent the change in population, the population size at t times t plus 1 can be represented as the initial population at time t plus additions that are caused by births and immigration, and deletions in the populations which are caused by the processes of deaths and emigration. Often when we are studying a population in the wild, we are interested in the rate at which the population is changing. So we'll begin with this equation which shows the population at time t plus 1, and let's for simplifying this equation, let's just ignore migration. So basically we are assuming that the population is geographically closed and doesn't allow movement of individuals. So individuals can't come from outside, neither can individuals of our population can they move out from this population. So now the equation becomes simpler. In at time t plus 1 equals to the initial population size nt plus the difference between births and deaths. Because we are interested in rate, so what we do is instead of looking at absolute number of births and deaths, we derive births per capita, which is given as letter small b, which is given by this equation b equals to capital B, that is the total number of deaths divided by the initial population size. And similarly, we can also derive deaths per capita. Now what we do is we can represent the same equation as n at times t plus 1 equals to the initial population nt plus a product of nt and the difference between births and deaths. And by dividing this entire equation by nt, we derive this rate parameter denoted by lambda, which represents discrete population growth rate and is given by 1 plus the difference between per capita births and deaths. If we are interested in computing or looking at the continuous population change or the rate of continuous population change, we start with the same equation, and we first look at the change in population size between these two time periods given by delta n, and that can be given as the product of nt and the difference between per capita births and deaths. And to look at this change over very small time intervals, we can take the differential of this equation, so we can get dn by dt, which is given by n into the difference between births, per capita births and deaths. And by dividing this entire equation by n, we can get something called R, which represents the intrinsic population growth rate, and this is given simply by the difference between births and deaths. So now we have two rate change parameters to look at population change, and lambda and R, and here it is represented how they would look like if they are plotted. So when n is on a plot with n changing with time, we can see that R, being continuous rate of change, is smoother than lambda, which is a discrete rate of change. Mathematically, R can be derived from lambda by taking the natural logarithm. Now let's look at how these two parameters are used in inferring how a population is changing when we are looking at a population in the wild. So for instance, if population is increasing, we infer this if R is greater than 0 and lambda is greater than 1. If population is stable, R is equal to 0 and lambda is equal to 1. And if we find that R is less than 0 and lambda is less than 1, our inference is that the population is in decline. Let's look at this using this example. So in this plot, we see that population growth rate has been plotted against the proportion of non-native invasive plants in the habitat. And we see for these Carolina chickadees, we see that the population, the R, the intrinsic rate of population growth, it declines very rapidly as the amount of invasive non-native plants increases in their habitat. As we can see, this red line is steadily declining. If the population was steady and not changing, we would have expected this line to coincide with this green dotted line, which represents how the R value should be if it was stable. Population change can also be examined in a slightly different way. We can we can look at from time t to time t plus 1, what is the proportion of individuals that have survived. And when we are doing this, we usually look at the population, the adult members of the population, and this is something which is referred to as the annual survival parameter, which is represented by the Greek letter phi, and it is given by n at time t plus 1 minus the contributions due to births and immigration, and this whole difference is divided by the initial population size. So this gives you the annual survival parameter. Similarly, we can also compute recruitment, which is the per capita additions that have happened in the populations from time t to t plus 1, and this is given by the small letter f, where n is given by the the ratio between the population at time t+1 by the population size at time t, and from that we subtract phi to get recruitment. This is an example study which used this survival parameter to look at how morphological and behavioral traits of species influence if they are impacted by selective logging. If you look at the first plot A, you see that the survival parameter doesn't change much among birds which live in intact forests and logged forests with increase in body size, and we can infer that due to this overlapping confidence intervals. Next, in plot B, we look at the social behavior, how social behavior, and then in plot C, we look at migratory behavior, how these two traits influence whether bird species are impacted by logging. We find that there is no difference in survival for species that participate in mixed species flocks and those which show migratory behavior. So for birds with these traits, there is no difference in survival between intact forest and logged forest. But for birds which are solitary in nature and do not participate in mixed species flocks and which stay in the habitat through the year, so the resident species, these are the two groups which show a decline in survival as we move from intact forest to logged forest. So these are the behavioral traits for these bird species which make them vulnerable to selective logging in the eastern Himalayan habitats. So this is one way we can use survival to look at influence of certain habitat disturbances on bird species. Another parameter which is used very often to look at population change as well as how habitat is affecting populations or habitat changes or disturbances affecting population is population density. We see it very often because when compared to computing rate change parameters like small r and lambda, which require at least 3 years of data to look at and compute these parameters of interest with sufficient precision, population density can be computed or estimated much easier. You can just visit a site one time and using some appropriate sampling methodology such as line transects or point counts, you can estimate population density quite reliably. So given this ease, you see this in literature much more often. So population density basically refers to the number of individuals per unit area, and for bird studies we often see it being expressed as the number of birds per square kilometer or hectare, and it is used to both understand trends in populations, so to look at change in population over population size over time, and by comparing population densities across different habitat types or disturbance regimes, it is also used to see how these changes are affecting populations. So there are some standard methods which are used to estimate population size, which will be addressed in a subsequent lecture. Now I have exposed you to a bunch of population parameters that are used to look at changes in population size. The first three, that are population size, the density, and another metric which is often used, which is the proportion of patches that are occupied by the particular population of interest, which is also referred to as occupancy. These three are usually termed as state variables. What it means is that these three parameters tell you about the current status of a population at a particular point in time, and state variables basically by the very nature change with time. The next set of parameters, which are births and deaths per capita, the annual survival parameter, recruitment rate, and the two rate change parameters lambda and r, are referred to as vital rates, and they are different from state variables because while state variables just tell you about the current status of populations, vital rates refer to the processes that make change to the state variables. So they are much more informative, and of course they are much more difficult to estimate in field because they need repeated years of monitoring to derive these estimates with with enough precision. So let's just look at how these two parameters make why are they important, and what are the difference between the inferences that we can draw by using these two types of parameters. So here is an example which looked at changes in firstly they looked at changes in density of white-bellied shortwings in the sky island habitats in southern India, which are naturally patchy or naturally disconnected in nature. So when they looked at density of these birds in plots in large forest patch and plots in multiple small forest patches across a disturbance gradient, what they found was that the density was more or less similar across all the patches in the large forest patch. So the densities were not very different, and they didn't show any discernible trend. So less disturbed plots had lower densities than more disturbed patches, and the errors were quite overlapping. So we don't see any discernible trend when we look at density. But when you looked at the survival across these patches, there was a very clear trend. So as we moved from more disturbed to less disturbed and subsequently to undisturbed habitats, we saw that there was a gradual or rather steep increase in annual survival parameters, showing that birds do much better in undisturbed habitats in both large forest patches and small forest patches uh, uh, as compared to more disturbed habitat patches.
Now let's look at why it is important to consider vital rates such as birth rates, death rates, survival, recruitment, and growth rates when we are monitoring ecological populations, and why it might be misleading to rely only on measures of abundance such as total abundance or density in making inferences on how bird populations are doing and how different habitat changes or threats are affecting them. Let's take this hypothetical example. As we move from the left to the right, disturbance is gradually increasing, so it represents a disturbance gradient. We are moving from a large, natural, relatively undisturbed patch to a more disturbed patch, and finally, at the end on the right side, we are in a monoculture commercial plantation. And in this example, what we are trying to understand is how this one particular bird species, which is known to be threatened and is of conservation importance, does as we move from this forested habitat to this more disturbed plantation. And as you can see, there are some distinct differences in these two habitats. The forested habitat has got, relatively intact canopy, a good vertical stratification in the vegetation. On the other hand, when we move to the commercial plantation, we see some mid-sized trees, but the understory is almost non-existent. Given these changes in the habitat and difference in disturbances, let's look at how we can go about measuring the impact of these changes on this particular bird species. So what we can do is identify two sampling units, uh, uh, two cells, and what we do is we estimate the density of this particular species of interest in both these grid cells. And what we find and we do this over 3 years. So when we look at the results in this hypothetical example, we find that year after year we get a density of 6 bird per unit area. On the other hand, when we look at the plantation, we find that we get a density of 4 birds, which again remains unchanging across 3 years. So, based on a comparison of density alone, we might say that these plantations are able to or show densities that are lower than what is seen in the intact forest, but it seems to be more or less stable across years. So based on this comparison of density alone, you might be very tempted to say that these commercial plantations do a relatively good job of conserving this particular bird species. But we to really make that inference, before we can make such an inference, we need to look at the data more carefully, and this is what we're going to do next. So now what we are overlaying on the density data is data on survival. In this case, we can look at the birds, and as we move from year to year, we see that in the forested habitat there is very little turnover. So from the first year to the second year, there is a small only 1 out of 6 birds is different, and from the second year to the third year, now there is one more bird, 1 out of 6 bird, which is again different. So this is a very low turnover across years. So most of the adults are actually surviving between years, and there is very little change, which is most probably brought up by birth or recruitment or migration of individuals into this population. On the other hand, when we look at the population from the plantation, we see a very high turnover. So we are seeing that 3 out of 4 birds in the second year are different from what we saw in the first year. Again, similar turnover happens between the second and third year. So this results in the intact forest showing very high survival value. On the other hand, the plantation is showing very low survival values. Now, why does this happen? So typically, these kind of large natural forests which are relatively undisturbed, they act as source populations, which are characterized by either stable or growing populations, and typically they because the population tends to be increasing over time, these habitats tend to pump out surplus individuals, so they are known as net exporters. So these additional individuals then go on to try finding habitat and territories outside this area, and this results in and what these surplus individuals end up doing is just go and occupy any habitat that they come across in the immediate vicinity, like this plantation for this in this example. But what we see is that given this habitat shows a lot of disturbance, or doesn't have a very good diversity of habitats which can support breeding or even provide good amount of resources for this particular bird, the dispersing birds which are trying to settle in this area, what happens is that either these birds die out or they move out of this area because they are not able to persist in this population or in this habitat. Such habitats which show a declining trend or a stable population, if the individuals it loses out are being constantly compensated from a nearby source, these kind of habitats are known as sink, and they are characterized by a very high turnover in individuals across years, because every year a certain number of individuals, surplus individuals, are arriving from the source. Then in the next year, most probably they are dead or moved out, and a new bunch of surplus individuals have then come to occupy this habitat, which is able to maintain the density, but actually such a habitat is not capable of supporting a growing population or a population that can sustain itself in the absence of migration. So this population is being almost completely supported by dispersal happening from a nearby source. So that's why when we are trying to make any inference about if a particular habitat management or a threat influencing a population of wild birds, we need this kind of data on vital rates such as survival, growth rate, to be really make an inference about if that habitat or that practice is really helping or supporting the population that we are interested in.
Another interesting approach that is used for looking at demographic parameters or population parameters is known as life tables. This is not very commonly used because you need very detailed data on a particular population of interest to use live tables to compute population parameters. But in any case, it has been done for some very intensively monitored populations, such as certain populations of Darwin's finches in the Galapagos Islands. What live tables basically contain is distribution of how many individuals are there of a certain age across the different age classes of that species, and it also has information about age-specific birth rates or fecundity. So as you can imagine, such information is rather difficult to collect in nature and we hardly come across it. How do we calculate population parameters using a live table? Let's look at that. So we can first calculate something which is called as survivorship using this formula. So basically survivorship is given by number of individuals that are alive at a particular age divided by the initial number of individuals. So basically every Sx value divided by the initial number of individuals, which is 100 in this case, gives you the survivorship for that particular age of interest. The next, we can calculate something which is known as the age-specific survivorship. So what is the survivorship at every age class for this particular species? And this we can compute by dividing the number of survivors in the next age class. So for age equal to 1, you look at the number of survivors in the next class, which is 12, and divided by number of survivors in that particular age class, which is 12 divided by 25, which gives us 0.48 in this particular case for age class 1. Next, you can also calculate something known as the net reproductive rate, or R0, which basically is given by firstly taking the product of survivorship and age-specific survivorship for every age, and then we sum these values. So for this particular hypothetical example shown here, this value comes up to be 1.29 offspring. So the net reproductive rate basically tells you how many, on an average, how many offspring does a female produce in a particular population of interest. We can, using the same data, we can also calculate the average generation length for that species given by T, which is equal to the product of X, which is the age, and LX and BX, which is the survivorship and age-specific survivorship, and this value is summed across all the age classes and divided by the net reproductive rate gives us the average generation length, that is the age at which a species starts reproducing. It's also, this also tells us the time at which a turnover of generation happens in a particular species. This is very informative for species. For instance, if you look at critically endangered species in our country, such as the great Indian bustards and white-rumped vultures, which have suffered a huge and drastic decline in recent years, they typically have very long generation lengths. For great Indian bustards, it's 15.6 years. For white-rumped vultures, it is 11.3 years. When we compare it to generation lengths of relatively more common species such as Indian peafowl and Asheprinia, we can clearly see that the generation length is much lower for these species. So generation length is a very important criteria which influences the conservation status of species. And also it also influences, if you are trying to recover an endangered species, the longer the generation length, the more time it is going to take us to revive the species from decline. Another very important parameter, which we already estimated in the previous slides, is the intrinsic rate of population growth, small r, that can also be calculated using live tables, and here is the equation for doing that. So r can be expressed as the natural logarithm of R0, which is the net reproductive rate, and this, when divided by the generational length of the species, gives us the intrinsic rate of population growth. So this is how we can get many population parameters of interest if we have access to a good life table. Now let's look at how population grows in nature and what are the factors that regulate such population growth. So usually population growth is described using two idealized models of population growth known as exponential growth and logistic growth, and in the subsequent slides we'll look at them in turns. Population growing exponentially, it basically grows very rapidly till resources are readily available in the habitat. Such populations often crash abruptly when the resources are exhausted. And this kind of population growth is mathematically represented using this equation, which is Tn, rate of population change, is equal to a product of intrinsic population growth rate and the initial population size, which we have seen this equation when we derived R. So this is how it looks when we plot exponential growth rate, and we can see that the higher the R, the higher the value of R, the steeper your population growth rate curve is. Here we can see that when the growth rate is 1 as compared to 0.5, this is how much steeper it gets over time. One of the most well-known examples of population growth is the recovery of whooping crane population in the United States of America. So whooping crane remains endangered to date, and its population was reduced to a mere 22 birds in 1967 due to the combined effects of hunting and habitat destruction. But subsequent conservation measures have resulted in this population increasing rapidly over time because the remaining habitat and resources that were available in that habitat were more than enough and readily available for this meager population to grow rapidly. Currently, there are close to 480 birds in the wild, of which 250 are mature birds capable of breeding. So it's still on its trajectory to increase. But in nature, hardly any population can grow exponentially forever, and that is because resources in any habitat are finite or limited, and as populations increase, they have to compete for the resources available in that habitat, and that limits the number of individuals that can occupy a particular kind of habitat. This maximum population size that any habitat is capable of supporting is referred to as the carrying capacity of that particular habitat and is denoted by the capital letter K. We must note that K is not fixed. It can vary across space and time as habitat itself changes, and as a result of it, the resources available in the habitat change. So the mathematical expression which shows logistic population growth is shown here. It is just the product of R, which is the intrinsic population growth rate, with the initial population sizes in time t, and this is then multiplied by this additional parameter, which is 1 minus the population size divided by carrying capacity. And this term basically refers to the environmental resistance, or rather the resistance offered by the habitat as population grows, and this resistance tries to keep the population size within the carrying capacity. So the higher the n, the more is the environmental resistance offered by the habitat. And because of this property, we see some distinct phases in the when we plot logistic growth over time. When population is small and n is closer to 0, this value of k minus n by k becomes 1, and we can see that the population at this phase grows very rapidly, almost mimicking exponential growth, and this is referred to as the logarithmic phase. Subsequently, in increases or at larger values of n, where it tends to be very close to k, this entire term becomes 0 and your growth stops, and this phase is known as the negative acceleration phase. And based on these properties of environmental resistance, two distinct classes of factors have been identified which affect or regulate population growth. The first class of factors are the density dependent factors, and the influence of these factors is actually mediated by the density at which the population exists at. So these include diseases, competition for resources, effect of predation, parasites, and stress. And all the influence of all of these factors tend to increase as the density of population increases. The second class of factors that influence population growth rate are unrelated to density, so their impact does not depend on the number of individuals that are present in a particular habitat. And these include factors such as natural calamities, which could include hurricanes, fire, flood, extremes of temperature, and all of these act on a population which is in a manner which is unrelated to the density of individuals. So density dependent factors typically affect the population by acting on births and deaths. So when individuals of the same species within a population compete for the same resources through intraspecific competition, it tends to increase as the population size increases, and this might decrease birth rates or increase death rates, and this results in decline of the population. So this is how it works. And here's an example of density dependent factor affecting population growth rate. In this example, we see that these tits, their average clutch size declines as the population density, indexed in terms of number of pairs that are present in a hectare, increases. So this might happen because the resources that are required by a mating pair to nest and produce the clutch will decline, as the population density will increase in that particular habitat, and as a result of it, we see fewer and fewer clutches, fewer and fewer number of eggs being laid by the remaining mating pairs. Here is an example of density independent factor regulating a particular population. Here we see that the population increases and decreases periodically, and this has got nothing to do with the number of individuals that are present at a particular point of time. And this is an example of song sparrows found in Mandate Islands, and here they saw that the population crashes abruptly from time to time in response to severe winters. Another example of population growth which does not correspond to either exponential or logistic growth are these periodic boom and bust cycles characterized by rapid population growth followed by steep declines, as shown by these crossbills in Norway. And they found that this was a response to similar crash and boom and bust cycles, which was shown by their main food supply, which was the spruce crops. These birds consume these cones, and their population corresponds with the cycles that are shown by the spruce crop in that particular habitat. Another information that we often look at when we are studying populations is how are individuals distributed in space, and this is referred to as population dispersion. So there are different ways in which individuals might be distributed in space. They might be showing clump distribution where individuals are aggregated in space, and this is the most commonly encountered pattern in nature, and there are multiple reasons why individuals might be aggregated in space. Firstly, there are certain habitats which are more suitable than others because habitats are not homogeneous, they are heterogeneous in nature most often, and as a result of certain habitats being more suitable, individuals might aggregate to occupy those habitats. Similarly, Resources are also not homogeneously distributed, but are rather distributed or more abundant in certain patches, and individuals tend to aggregate to capitalize or utilize those resources, patchy resources, as they are found in nature. Then there are also certain social factors, like offsprings usually tend to stay with parents. Individuals might also aggregate to come in contact for mating purposes. Then, so individuals come together and forms flocks to escape from predators. And if you look at the distribution of any species at a large enough spatial scale, you'll find them to be clumped in nature, because the habitats that are suitable for them won't be widely distributed in a uniform fashion. So that's the most common form of distribution that we see in nature. One of the most visually obvious clump distributions, which all of you must have seen in recent times, is the distribution of Amur falcons in the wintering stopover site in Nagaland. Similar, aggregated distribution or dispersion is seen along the migratory pathways of many birds like snow geese in this particular picture, and this is usually the sites where they stop over on their migratory pathways to rest and refuel. Then the second type of dispersion that we see in nature is uniform distribution, where individuals are evenly spaced in a particular area, and this is often seen among territorial species, which basically try to capture a certain area where resources are limited. So individuals compete for those limited resources, and they maintain these territories, and these are usually maintained by antagonistic behavior or interaction. So they fight with each other, or to maintain their particular territories, which by themselves contain enough resources. So here is an example, a visual example of such uniform distribution of nesting colony of gannets, and these are not as common as clump distribution, and we find them rarely. The third type of distribution or dispersion that we see in nature is random distribution. Here, and these are very, very rarely encountered in nature. Here, individuals have equal probability of occurring anywhere in a particular area that is suitable for them. And the assumption behind this is individuals interact with each other in a neutral manner. And similarly, the interactions between individuals and resources are also neutral. And resources have to be randomly or uniformly distributed for such random distribution to occur. One rare example that we see in nature of uniform dispersion is the distribution of dandelions or similar wind dispersed seeds. Here we can see that as long in the habitat that is suitable for this particular species, wherever the seed lands, a plant can germinate and grow, but this is very rarely encountered in nature. Now that we know about what are the patterns of dispersion that we usually see, how do we discern which pattern it is when we encounter a particular population? So there are ways of doing it. We can actually measure and find out which particular dispersion it is when we encounter a population. So here's an hypothetical example. So we are laying 16 plots in a particular area, and in each of these plots we can count the number of individuals of a particular species. We have done that. We have found that 12 of these are empty, and only 4 cells have 10 individuals each. By just looking at it, we can say that it's a clump distribution, but is there a mathematical way of confirming if that is indeed the case? So Krebs in 1999 came up with a measure to tell us what kind of dispersion it is, and this is known as the variance to mean ratio. For this, we first calculate the mean for this particular distribution that we have measured. In this case, it is 40 individuals divided by 16 plots, which is equal to 2.5. We then calculate the variance for this population using this formula, and here it comes to be 20. Then we compute the variance to mean ratio, which is 20 by 2.5 in this case, and it equals 8. According to Krebs' rule for this, for discerning the population dispersion, if the ratio is more than 1, we consider it clumped, which is the case here. If it is less than 1, it is to be treated as uniform, and if it is equal to 1, we consider the dispersion to be random. Subsequently, many more sophisticated measures have been developed to identify what kind of population dispersion a particular population is showing, but the basic principle remains the same. So this brings us to the end of the lecture on bird populations. In a subsequent lecture, you'll be learning in detail about how we measure all these parameters in nature using field studies. Thank you so much.